Welcome back to the second instalment of the Bridging series designed to help you prepare to start your A-level biology journey. In this video, we are going to give you a head start to ease the transition to A-level by consolidating core GCSE knowledge needed for Year 12, and we're also going to throw in a few nuggets of A-level knowledge in advance this time on the topics of cells, transport, and the immune response. All of these topics are studied in all exam boards at A-level. So before we start, do pay attention to the free downloads in the description, including one on approved abbreviations from AQA. Having this in the front of your folder for reference would be really useful and prevent you from losing marks early on from using unapproved abbreviations. So let's refresh core knowledge to strengthen our understanding on cells. Your GCSE fundamentals should mean you are confident on the parts of eukaryotic animal and plant cells and what their role is. Feel free to pause the video if you need a quick review using the summary table. The most commonly forgotten are the little ribosomes. Because they're so small, they are the really important site of translation in protein synthesis. And remember, plant cells have three extra structures not found in animal cells: the cell wall, the permanent vacuole, and the chloroplasts. You will learn so much more about the ultrastructure of some of these organelles, including the mitochondria and chloroplasts, and how they perform vital cell functions. You should also recall the structure of bacterial or prokaryotic cells. The biggest difference being the lack of a nucleus or any membrane bound organelles like mitochondria or chloroplasts. Now, I still find my A level students wrongly assume that all structures are present in all bacterial cells, and it's important to get this secure early on. Components that you will find in all prokaryotic cells are the cell membrane, the cytoplasm, the ribosomes for protein synthesis. However, you will learn in A level they are slightly smaller. There's a cell wall, but it's made of a different substance, murin, compared to plant cell walls, which are made of cellulose. All prokaryotic cells have DNA, it's the same chemical structure, but its larger scale arrangement is different in prokaryotes in that it exists as a loop rather than distinct chromosome pairs. It's also loose in the cytoplasm and not in a nucleus. The most common mistake students make is thinking plasmids, flagellum, pili, and the slime capsule are in all prokaryotes. However, they're actually only found in some. So we know all of the detailed structure of cells due to developments in microscopy. You will have learned at GCSE the differences between the electron and light microscopes, and you'll extend this knowledge at A level. So let's remind ourselves. Important to emphasize first is some important terminology. Don't confuse magnification with resolution. Magnification is how much bigger the image appears. We can't talk about zooming in. Resolution is the ability to distinguish two close points as separate. The distance between these distinct points is the resolving length. And the higher the resolution, the more detail you can see because the resolving length is low. You'll have learned about two main types of microscope: light microscopes and electron microscopes. Light microscopes use light, can observe living cells, and produce colour images. They magnify up to times 2000 but have a lower resolution. We cannot see very small structures like ribosomes using them, but they can produce colour images of living cells and are relatively cheap. Electron microscopes, however, pass a beam of electrons rather than light through the specimen. They have a much higher magnification and resolution, revealing tiny structures like viruses. However, they are expensive. Specimens must be dead because they require a vacuum, and the images are in black and white. You will go on to learn about scanning and transmission electron microscopy at A level, as well as learn about some other important structures found in cells, such as the Golgi apparatus and the endoplasmic reticulum. Now, a huge point of emphasis I need to make is that you must practice labeling cell structures from electron microscope images during your revision. Examiners love an electron microscope image as students find them tricky, as they are used to lovely, clear scientific diagrams. On the topic of scientific drawings, a core skill examined at A level are the fundamental rules of scientific drawing. And these rules are from GCSE and still apply at A level, such as using single continuous lines with no gaps or overhangs, no sketching or shading, clear labels that don't cross, This is a skill on all exam boards' apparatus and techniques list, so make sure you know these rules or could apply this knowledge to improve or produce a drawing. There should be an indicator of scale of measurements in the form of either magnification size or a scale bar, which can be used to calculate the magnification. This is really commonly missed out on drawings students do at A level, so make sure this isn't you. I want to also review how to perform magnification calculations, as this is a GCSE skill that I see A level students still struggling with every year. So get this mastered now to boost your grades early on. Hopefully, you remember the equation actual size equals image size divided by magnification. I call this the AIM equation, as when it's in an equation triangle, this reads left to right, AIM. Some remember it as I am, reading top to bottom. You do have to recall this equation independently. You don't get an equation sheet. And for more information on maths equations you will need to learn and recall throughout A-level biology, see my video linked in the description. Now, when using this equation, remember the fundamentals of using any equation. Identify your knowns and unknowns, substitute into the equation, and convert units before carrying out the calculation. You must be secure on unit conversions, which I've summarised here. Now, a reminder what we do if we have a scale bar instead of a magnification value. On our scale bar, the length of the line, measured using our ruler in millimetres, is the image size. The number next to the image bar tells us what that length represents in terms of actual size under the magnification. So if we use the example shown and I line up my ruler against the scale bar, the image of the scale bar is 18 millimetres, and this represents an actual size of 25 micrometres. We convert 18 millimetres into micrometres by multiplying by 1000, and then we divide this image size by the actual size of 25 micrometres to get a magnification of times 720. We can then use this magnification to measure the diameter of the nucleus. So if I measure my nucleus with my ruler, the diameter is approximately 24 millimetres. If I convert this image size into micrometres and divide by the magnification times 720, I've calculated the actual size of the nucleus as 33 micrometres. So just remember, converting to a larger unit, the number will always get smaller as there will be fewer of them in the same space, so you must divide by 1000. Converting to a smaller unit, we will get a larger number in the same space, so you must multiply by 1000. I would avoid measuring in centimetres as you aren't able to use the factor of 1000 in your conversions, and students make mistakes more often. The same can be done for volume as shown in the diagram, and do remember that 1 centimetre cubed is equivalent to 1 millilitre. Let's now move on to transport across membranes, and I'm going to give you an advanced look at the detail in membranes you will learn about at A level. You can see the membrane is actually made up of 2 layers called a phospholipid bilayer, and we have proteins embedded in the membrane throughout. You should be familiar with carrier proteins from GCSE and remember that these can move molecules against a concentration gradient from low to high concentration using energy in a process called active transport. You'll learn at A level that proteins are also used for a process called facilitated diffusion. Remember simple diffusion from GCSE, the movement of molecules down a concentration gradient from high to low. It's a passive process that doesn't need energy from ATP. Now, some molecules can directly diffuse across the membrane. However, some molecules need to use proteins to cross the phospholipid bilayer in a process called facilitated diffusion. You'll learn all about which molecules use which type of diffusion and why. You should also recall osmosis, the movement of water across a partially permeable membrane from high to low concentration. You'll learn at A level how this can happen directly between the phospholipids or using special proteins called aquaporins. And you will repeat at A level the practical you will have completed at GCSE using potato cylinders and different concentrations of solutions. This will be one of your required practicals, so I'm going to re-emphasize the important points about this practical because you could very possibly be answering exam questions on it in the not too distant future. Our independent variable we change is the concentration of solution. Our dependent variable is the outcome we are measuring, which is the percentage change in mass of the potato cylinder. We've had to control the volume of solution, the time, the diameter and surface area of the potato cylinder, the age and species of potato, as this could affect the concentration of solution inside the cells. We record the starting mass of the cylinders and place them into solutions for a set time. We then record their final mass, but only after blotting them to remove excess water to ensure our measurement is accurate. We then calculate percentage change in mass to account for any slight differences in starting mass and provide a more suitable comparison. We calculate this by taking the final mass, subtracting the initial mass, and then dividing this change in mass by the initial mass. Then we multiply by 100. If we get a negative value, there has been a loss in mass as the concentration of the solution is higher than the cells, or hypertonic, so water has left the cells by osmosis. A positive value indicates an increase in mass due to water from a more dilute or hypotonic solution entering the cells by osmosis. Hopefully, you'll be familiar with a graph similar to this. We can estimate the concentration of the solution inside the potato cells by looking for where the line of best fit crosses the y-axis. This indicates the point at which the solution was isotonic, the same solution concentration as inside the potato cells. So no net osmosis occurred, so no change in mass was observed. So let's move on to our final topic of this video, the immune response, so that you're ready for the jump to A level. This is a topic where students find the jump significant. There is a lot of terminology and processes occurring in sequence, which, if we are not secure on the GCSE, can be very tricky to build further detail onto. Fundamental to this topic is revising key definitions and drawing process maps, so let's do this for our GCSE recall. You've learned about antigens, which are proteins on pathogens that are seen as foreign by the immune system. You should also recall that white blood cells called lymphocytes will recognize these protein antigens and release specific antibodies complementary in shape to the antigen. They will then bind to the antigen and agglutinate, which means clump together, the pathogens, which allows our phagocyte, another type of white blood cell, to engulf and digest the pathogen. Some of the lymphocytes will remain in the blood as memory lymphocytes, which will recognize the same pathogen in the future and destroy it quickly before symptoms develop. Vaccination aims to stimulate an immune response by injecting a dead pathogen or even just its antigens into the body to trigger the production of specific complementary antibodies and memory lymphocytes without giving us symptoms of the disease. We call the process from first exposure to the antigen to development of memory lymphocytes the primary immune response. We can see the presence of memory cells by the fact that the antibody level here does not go back to zero after the primary immune response. A second exposure to the same antigen will produce a faster and larger antibody production due to the presence of these memory cells circulating in the blood as a result of the primary response. The primary response is either due to first exposure to the pathogen or the antigen during a vaccination. If enough of the population have vaccinations, we can develop herd immunity, whereby unvaccinated have a level of protection as the pathogen is killed so quickly by those who have been vaccinated that there is very little spread of the pathogen within the population. Of course, there are many reasons why vaccination programs might not be successful, including mutation of the pathogen, lack of uptake of the vaccine, or lack of infrastructure or finance to deliver enough vaccines within the population. Probably the hardest part of the immunity topic at GCSE and also at A level is the use of monoclonal antibodies in diagnostic tests like pregnancy tests and to deliver cancer drugs to specific cells. Monoclonal antibodies are millions of copies of one type of antibody, generally produced from injecting mice with an antigen to produce many copies of the specific lymphocyte that is able to produce the desired monoclonal antibody. If we extract and fuse these lymphocytes with a tumour cell, we get a cell which is very good at dividing and producing a specific monoclonal antibody. If we attach a cancer drug to our monoclonal antibody, the specific nature of the antibody means it will only deliver the toxic drug to cancer cells which display foreign antigens on their surface. In this way, fewer healthy cells are killed by the cancer drug and symptoms are reduced for the patient. In pregnancy tests, we attach a coloured bead to the monoclonal antibody, which is complementary to the structure of a hormone found in pregnant women. If present in the urine, it will bind to form an antigen antibody complex, which will travel down the pregnancy test strip and then bind to an immobilized monoclonal antibody on the test line, producing a blue line in pregnant women. I've put a link to a great short video showing the same principle in COVID test strips. A control line is also included, which includes a second type of immobilized monoclonal antibody complementary to the mobile monoclonal antibody that's labeled. This will bind if the person is pregnant or not pregnant and serves the purpose of ensuring the test has worked properly and that a false negative test is not produced. So that is our review of the key information from your GCSEs that you're going to need in A level on cells, transport across membranes, and the immune response. Consolidating this is so essential to prepare you for the jump to A level. So if there's anything in the video you feel a little unsure about, take some time now to go back to it. I always love Cognito and BBC Bite Size for GCSE revision, but you will likely have found others that you like. Join me for part 3, reinforcing GCSE content essential for A level on gas exchange, digestion, the cardiovascular system, and plant transport. Subscribe to get notified on release, and if you want to get started now, check out the Bridging Series Shorts playlist.